This video is a recording of the fourth webinar in the SGD webinar series titled Comparing Yeast Strains with SGD's Sequence Variant Viewer. Please enjoy. All right, welcome everyone to the fourth webinar in the SGD webinar series. My name is Kevin McPherson. I'm an assistant biocurator at the Saccharomyces Genome Database, SGD, and today I'm going to show you how to use one of SGD's latest tools, Variant Viewer. Now, before we begin, please feel free to ask questions anytime throughout the webinar. Just click on the Q&A button on the right-hand side of the BlueJeans page, and the SGD team will answer your questions as they come in. We'll also be having a Q&A session at the end, so please be sure to stick around for that if you have any more questions. So what is Variant Viewer? Variant Viewer is an open-source web application created by SGD that enables users to compare nucleotide and amino acid sequence differences for a given open reading frame between 12 different S. cerevisiae strains. These 12 strains include the S288C reference strain and 11 alternative strains that have a substantial history in S. cerevisiae research, like W303, Sigma, and SK1. Variant Viewer can also be used to compare similarity scores between multiple open reading frames at once for all 12 strains, which is a feature that distinguishes it from other applications. To access Variant Viewer, open the SGD homepage at yeastgenome.org, enter the Sequence tab in the purple toolbar that runs across the top of the page, and expand Strains and Species. The link to Variant Viewer is inside, as indicated by the red arrow. Alternatively, you could just simply type in the URL, yeastgenome.org/variant-viewer. So let's go ahead and open up Variant Viewer. The Variant Viewer graph is shown on the left here. The x-axis of this graph shows the 12 strain names, while the y-axis shows the open reading frames being compared. To get started, simply search for a gene in the search box, like ECM22. Then, click on the gene to compare sequences. A map will be produced that indicates the location of any single nucleotide polymorphisms, insertions, or deletions. Hover over any of the lollipops to highlight the columns where the nucleotide change is shown. For example, here we see that at nucleotide 126, strain S288C encodes a cytosine, while W303 encodes a thymine. Moving over to the next lollipops, we see that there is an insertion at nucleotide 338. While S288C lacks this insertion, as shown here, strain W303, among other strains, has it. Something to keep in mind is that in Variant Viewer, synonymous and non-synonymous single nucleotide polymorphisms are shown. On the left-hand side of the page, click on the legend icon to see which each of the lollipop icons mean. If you'd like to compare amino acid sequences instead of nucleotide sequences, check the protein option. This will draw a map of the amino acid sequence and the predicted protein domains, which you can hover your mouse over to find a link to that predicted domain for more information. As before, hover your mouse over any of the lollipops to track the column that shows the sequence change. Here we see that at residue 113, the insertion that we saw earlier while viewing the nucleotide sequence is shown. Strain W303 has a glutamine insertion, where strain S288C does not. If you'd like to save a picture of the map you're viewing, on the left-hand side of the page, just click on the Download button. By the way, you don't have to limit yourself to just one gene in your searches either. Just add a comma in the search box and enter in another gene name. One advantage to Variant Viewer is that you can visualize high-level differences in multiple genes by using the matrix here. In this matrix, each of the 12 strains has a square that indicates sequence similarity to S288C. The lighter the square, the more similar it is to S288C. The darker, the less similar it is. For example, here we see that strain sigma has a more similar PIR3 sequence to S288C than does strain W303. It's important to note that in variant viewer, the white squares indicate that no comparison could be made, either because the sequence for the gene being compared is not yet available for that strain, or because the strain lacks the gene entirely. Lastly, a cool feature in variant viewer is the dynamic dendrogram, which is shown here. It indicates clusters of sequence similarity and is redrawn whenever you modify your search. So now that we've covered basic variant viewer usage, let's go over a quick research scenario. Let's say that you're interested in the fact that strain S288C, the reference strain, cannot undergo pseudohyphal growth, or a pattern of cell growth that occurs under nitrogen starvation, where cells become elongated and fail to separate during mitosis. Strain sigma, in comparison, undergoes pseudohyphal growth readily and is pretty well studied for it. A question you might ask is, what genomic differences between S288C and sigma, or if any, are responsible for the variance in pseudohyphal growth? You might postulate that S288C could possess some defect in genes that regulate pseudohyphal growth. To produce all the genes related to pseudohyphal growth in variant viewer, simply enter in the gene ontology term in the search box, regulation of pseudohyphal growth. This will produce a list of genes annotated to that gene ontology term, along with other genes that have matches to the words in my search. Since I'm interested in viewing strain sigma and strain S288C only, I can enter in the strains box, click on deselect all, and enable sigma again. Now, only strain S288C are shown in the dendrogram now. Since variant viewer runs its comparisons using S288C as a reference, S288C itself cannot be disabled. This is plenty of genes to sort through as it is, so I may want to reorganize this list. To sort by the level of variation, enter the settings box and check the variation option. This will reorganize the list so that the genes with the most differences are listed at the top. Here it appears that flow8 is listed at the top, so let's find out how S288C flow8 and sigma flow8 differ. I'll just go ahead and check the protein option since we're interested in functional differences. And here we are. Here we see that where strain sigma encodes a tryptophan, strain S288C encodes a premature stop codon and has thus likely lost flow8 function. Flow8 S288, the flow8 locus summary page on SGD indicates that flow8 is a transcription factor required for flocculation, filamentous growth, and invasive growth. Following this up with a quick literature search also reveals that the premature stop codon in flow8 is responsible for abrogating pseudohyphal growth in strain S288C. Okay, so that concludes our webinar. Thank you for coming, and please keep an eye out for our follow-up email on our next webinar and a link to our YouTube channel where today's webinar will be uploaded. Thank you for watching the SGD webinar series. If you have any questions or comments, please contact us at sgd-helpdesk@lists.stanford.edu.